Now we've made it all the way through taking our 1,6-carbon molecule and breaking it all the way down into our 2 3-carbon molecules. And now what we need to do, though, is think about this process, this whole pathway, and talk about the regulation of it. Now, when it comes to the regulation of these 10 different steps, steps 1, 3, and 10 are exergonic. They are metabolically irreversible, and they are tightly regulated. Steps 2, 4 through 9, so all of our other steps, are metabolically reversible. Okay, so they can be reversed. So there's, if we're going in one direction, those could go in the other direction as well. These are not control points. These reactions are very close to equilibrium. They are therefore very easily reversible. So steps 1, 3, and 10, those are the ones that when you go in that direction, it's a favorable direction. Going in the opposite direction is highly unfavorable. 2 and 4 through 9 are very reversible, and they are not something that is going to be controlled because they're close to equilibrium. Now, what that looks like is here are our glycolytic enzymes that catalyze irreversible steps. They are, and this numbering is a little bit weird. This is a sequence. I'm going to go ahead and scribble this out so that's not confusing. But the enzymes, rather than thinking about it in terms of numbered steps, think about it in terms of what enzyme is actually catalyzing a step. Hexokinase takes glucose and makes glucose 6-phosphate. So we've got a phosphorylation event that happens here. Phosphofructokinase is another kinase that is going to, again, phosphorylate a molecule. It's going to take fructose 6-phosphate and make fructose 1,6-bisphosphate. Pyruvate kinase is our last enzyme in this whole pathway, and that's going to take phosphoenolpyruvate, PEP, in these 2 different steps, and it's going to transfer a phosphate group from, well, PEP onto ADP. Now, this might seem a little bit strange because of the fact that, well, this is kind of removing a phosphate group from this phosphoenolpyruvate and making ATP. Shouldn't that be a phosphatase enzyme? Well, this is a pyruvate kinase, or this is a kinase as well. The reason for that naming, it being a kinase rather than a phosphatase, is because of the fact that it's lower energy, or it's making a higher energy molecule. This ATP is higher energy than phosphoenolpyruvate. All right, so those are our steps. So hexokinase, phosphofructokinase, and pyruvate kinase. Now, ultimately, when we produce that pyruvate, well, what's going to happen with that pyruvate? It's going to go in a couple of different directions. One, we can have the complete oxidation to CO2 and H2O, and that's through our citric acid cycle, which we'll get into next. That's going to produce a bunch of CO2, H2O, as well as NADH and protons. Okay. We could also have alcoholic fermentation. Now, that means that that pyruvate is going to be converted to CO2 and ethanol. The final output, or the final outcome, is what's known as homolactic fermentation, or fermentation to lactic acid, where we take pyruvate and we generate lactate. So these are the 3 possible outcomes of pyruvate. Now, when pyruvate is metabolized ultimately to our acetyl-CoA and enters the citric acid cycle, well, we're going to have a different output than the other fates. One of which it's completely oxidized to CO2 and H2O. This is an aerobic process. We have to involve oxygen in this entire process. Here is the total reaction. If you think about this upstream, well, it took 1 glucose to make these 2 pyruvates. These 2 pyruvates are paired with 4 waters, 8 NAD, 4 FAD, 2 GDP, plus 2 inorganic phosphates to produce CO2, or 6 CO2, 8 NADH, 8 H+, 2 FADH2, and 2 GTP. Our fermentation pathways are with the enzyme, our homolactic fermentation is done in the muscles. And this is done by an enzyme known as lactate dehydrogenase, where we're going to take pyruvate and NADH to produce NAD+ and lactate. Our alcoholic fermentation is going to take which is done in yeast, is going to take pyruvate. It's going to decarboxylate it to make acetaldehyde. It's going to dehydrogenate it to make ethanol using the enzyme alcohol dehydrogenase. So the biggest differences, or the questions that you should kind of ask yourself is, what's the fate of pyruvate? Well, you've got three fates, but is there an oxygen-rich environment or an oxygen-deprived environment? In an oxygen-deprived environment, you're going to have pyruvate converted to lactate. Now think about this in terms of, well, where's it taking place? It's taking place in the muscles. When you get that, when you're exercising, you get that lactic acid burn. Well, that's what we're looking at here. We're producing this lactate. Our concentration of lactate is causing this burn, or that unpleasant sensation to occur. Alcoholic fermentation, on the other hand, well, you're not producing any lactate, but rather you're taking pyruvate to make ethanol. So those are the three fates. We've got one that is taking place in an oxygen-rich environment, and two that are taking place in an oxygen-deprived environment. All right, well, that's the end of this stuff. We'll see you in the citric acid cycle discussion.